Great. Well, thank you so much. And thanks to the organizers for this invitation. I'm really happy to share our progress to date. And I also want to recognize that I'm probably sitting between you and either your lunch or your coffee break. So I'll try to keep this fairly high level, but I would encourage you, if this is of interest, to please reach out to us and to start engaging with this project, because I think this is a really exciting opportunity to get involved in this pilot program that we have across many different national labs. So I first wanted to go through what the NMDC is for those of you who may not be familiar with this pilot program. I'll emphasize the importance of fair microbiome data, work that we're doing in terms of making accessible workflows for processing a variety of multi-omics data, the infrastructure that we've stood up over the past 2 years, and then some of our partnership and engagement activities. So first, I wanted to start with just a statement on what the problem is and what we're hoping to accomplish with the NMDC. So I'm sure many of you are very well aware that there are a lot of challenges in scaling omics data, and all of the great advances that we've seen in terms of sequencing microbes in their natural environments, or using other omics technologies like proteomics or metabolomics to probe the function of those microbes in those communities, has really outpaced the current data infrastructure to support integration of these multi-omics data sets in a meaningful way. So this is a silly picture that I like to show just to give a visual perspective of what we're looking at in terms of the current scale of analysis in this little green cube here, and then the expected scale of analysis in terms of different taxonomic scales, ecological scales, or genetic scales over the next several years. And so what we currently have might be a number of different repositories that host these data that are providing very useful resources. But the challenge is, how do these resources talk to one another? How can this data be integrated or harmonized in a meaningful way to enable cross-study comparisons, to be able to look across different studies and be able to pull out patterns or processes that are meaningful outside of maybe the original study from which this data has been generated. And so the goal is to take that spaghetti of different data and sources and to try to harmonize it in a way that researchers around the globe can be able to access this data in a meaningful way and ask questions that wouldn't previously been able to be possible to address. So that's really kind of the guiding principles for what the NMDC is. Our vision is to try to empower the research community to harness all of this data into meaningful discovery through an integrative data science ecosystem. This is a pilot program funded graciously from the Department of Energy, BER, and we're a distributed research group across 4 national labs: the Berkeley Lab, Los Alamos, Oak Ridge, and Pacific Northwest. And we're focused on data standards, on the data quality, integration, and importantly, data access. So enabling all of these capabilities within the NMDC, but also across our partner organizations. So we're, as I mentioned, a partnership across 4 national labs. Here's a picture. I always like to show this of our diverse team. It consists of microbial ecologists like myself, but also software engineers and developers, and a whole range of people coming from a variety of different fields. And so I think this is really critical in terms of developing this community-centric infrastructure in a way that will be meaningful to use data across diverse domains of microbiome research. So I mentioned what our vision is. When it comes down to it, our mission is really to take diverse data, specifically from the Joint Genome Institute, from EMSL, and other resources to try to standardize those data in systematic ways, both in terms of the environmental contextual information or the metadata, as well as the instrumentation data that can be processed through workflows that have been standardized that I'll speak to in a minute, and then to enable all of those data to be integrated and accessible and searchable by researchers. So if you're interested in getting more information about the technical details of the NMDC, we have a bunch of information on our GitHub site in terms of the metadata and the data schema, as well as the available bioinformatics software components that are available as containerized workflows. And so I'll just provide a very high level description of all of the various components over the next couple minutes. Also, I wanted to mention that this pilot program really focused on leveraging projects that have been generated through the ficus program, or the Facilities Integrating Collaborations for User Science. And this is a great mechanism that through one proposal, you can get access to capabilities through both the JGI and EMSL. And importantly, all of the projects that have been generating data through the ficus program really provide a rich resource of diverse multi-omics data that we are beginning to pull in collaboration with those respective research teams to be able to host within the NMDC data portal. All right, so one key aspect that I wanted to mention is a huge emphasis on data standards. And we've been very fortunate to be able to work across a really nice range of community consortia and working groups that have been in the business of developing standards for many years now. And then importantly, to try to bring those standards together in a meaningful way to better support microbiome research. So we hosted a workshop now several years ago in person, which seems, I guess a bit foreign in our virtual world that we live in. But it was really nice to get together these various groups and to put forward ideas about how we can better coordinate and harmonize metadata standards across multi-omics data in a meaningful way. One aspect of the standards that we're using are standards called the MIX-S, or the Minimal Information About Any X sequence. These are standards that have been generated over the past several years by the Genomic Standards Consortium, and they describe various minimal requirements or components for describing the contextual information around specifically sequence data. And so we've been leveraging these standards, which have been very important in the field of genomics and metagenomics, and applying them more broadly across multi-omics data, so proteomics data, as well as metabolomics data. One aspect that I wanted to just very briefly share is that we've spent a whole lot of time over the past 2 years developing a robust data schema that leverages these standards, but then also integrates across data that has been generated at EMSL and at JGI. And so this is just a snapshot of a component of the data schema that takes into consideration the biosample information, but also the omics processing information and data, and puts it together in the context of the schema in a meaningful way to enable integration. And again, we're leveraging standards developed by the Genomic Standards Consortium. I also wanted to very briefly highlight that we have spent a lot of time and energy in developing standardized workflows. These are workflows that are in production at both the JGI and EMSL, but that have previously been restricted to the computational resources of those user facilities. And so what we've been able to do is to extract those workflows, develop them in a way using containerization and workflow documentation language to be able to have them be agnostic to the computational resources on which they run. And so you can go to our Docker Hub site or other resources. Eventually, these will be made available through KBase, and to be able to run these workflows in the same manner that data might be processed in the user facilities setting. And these workflows include processing pipelines for metagenomics that range anywhere from QC all the way to metagenome binning, metatranscriptomics, metaproteomics, and a suite of different metabolomics processing pipelines. And importantly, all of these workflows have been leveraging very similar components. And so the annotation of the data can be integrated together once data is processed through these workflows. I also wanted to highlight, I briefly mentioned that we're planning on working closely with KBase to enable these workflows to be made available. We currently have a beta version of the metagenomic workflows available in Edge. This is a system that is a very user-friendly platform developed by the Los Alamos group, part of the NMDC, where users can go to this web interface. The link is here if you're interested in testing it out, and be able to run any one of the metagenome workflows from the NMDC. This is currently hosted through HPC resources at the San Diego Supercomputer Center, and we plan on making available other omics workflows to be able to run all of the NMDC workflows through Edge at the end of our pilot phase. I also briefly wanted to mention that we are developing the NMDC using distributed infrastructure or distributed resources. And so instead of having data housed within one brick and mortar resource, we are instead having different access points at Berkeley Lab, and specifically leveraging the NERSC Supercomputing Center, but then also using EMSL's resources and HPC resources up at Pacific Northwest. And so this is all kind of seamless to a user who comes to the NMDC portal, but this is important in terms of where we hope to go for scaling the NMDC to petabytes of data in the future. And so what we rely on is a centralized metadata store, but when you go into the portal to make a download request, it triggers where you can access that data, whether at NERSC systems or at EMSL systems. So that's a little bit of the architecture for how we're developing the NMDC. I also wanted to highlight that we are very heavily focused on leveraging a community-centered design process. So what does that mean? So that means that we are very early on putting forward to the research community a prototype of the NMDC data portal that may not be perfect, but we hope that through working closely with research teams that we get input on the design, and that we can better plan and adapt certain functionality to really enable researchers to use the NMDC to best serve the purposes of their research. So instead of other systems that might be able to have a whole lot of data available and be able to enable only a certain or limited amount of functionality or capabilities, we're working very closely at a very early stage to develop new tools and new ways of accessing the data that will hopefully be more meaningful to the research community at large. So it was just this past spring or past March that we officially launched the NMDC data portal. Here's a snapshot of what that portal looks like, and the link to the portal if you're interested in looking at what data and functionality is available. We've also paired that release with quarterly newsletters that will be, again, sent out to the research community. So if you're interested in knowing what we're up to, I encourage you to sign up for our newsletter. And then I just wanted to very briefly run through some of the capabilities of the data portal itself. So here is the front page. It has a lot of information on it. We have focused a lot of time and effort in the design and development process, and especially talking with researchers to really focus in on how researchers want to search and access data. So we have a panel on the left side here where you can search based on environmental metadata information, or also enabling search based off of functional annotations, which I think is a really powerful way to integrate across multi-omics data and search based on functional annotations. At the top, we have information about our standards and our workflows, so you can dig into details for what how we're being developed and what partnerships we have. And then lastly, you can get information about the number of studies and search based off of studies or multi-omics information through these two components. So there's a whole lot of information just on this single web page alone. We do have a user guide that will be uploaded shortly to help in terms of navigation and how you would access data or search for data across a variety of different facets. All right, next I wanted to briefly talk about the ambassador program. So we have been working quite a bit on how best to engage with the research community and engage in a meaningful way across a variety of different researchers. So we've focused on developing an ambassador program where we recruit early career researchers to focus on developing training materials and gathering community feedback on metadata specifically. So this is a program that is not DOE focused, but is instead focused on a broad range of different domains and research communities in the entire microbiome space. And so we have onboarded 13 fantastic early career researchers pictured here. These early career scientists span a whole different range of research topics and institutions pictured here. And I would really encourage you to look at their biographies posted on the NMDC website. And they're gearing up in the next couple months to do some really exciting training activities as part of their involvement in the ambassador program. So if you see any of these training activities within your organization, I'd really encourage you to attend. It's really exciting that we have such a fantastic cohort of ambassadors who are really pushing on the importance of metadata and standardization, both in their work and more broadly within the research communities they work with. I also wanted to highlight that we have a really nice collaboration with the American Society for Microbiology. This has included providing material in the form of fact sheet to better socialize with the broader community the importance of microbiome data science. We were also really excited to partner with ASM earlier this summer to launch the very first award focusing on data stewardship and microbiome data science. And we're very excited to have Dr. Patrick Schloss be awarded this inaugural Microbiome Data Prize. We're also partnering with the ASM Ambassador Program for a lot of the training activities that the ambassadors will be involved with over the course of this year. And then lastly, I just wanted to mention we will be partnering with ASM for a coming November 15th congressional briefing series. And so you'll hear more about the NMDC, but also hear it from the perspective of interagency coordination, which I think will be really exciting. We've also focused very heavily on looking and finding ways to better support data management best practices. We've partnered with the DMP tool and developed a DMP template specific for microbiome studies. So I would encourage you to look at this. There's also a webinar that's interestingly enough going on right now. The recording will be made available, but I would really encourage everyone who is interested in data management and what the best practices are to look at some of our resources. I also wanted to mention that the NMDC is very invested in developing ways in which we can be more inclusive and equitable and importantly accountable to those diversity, equity, and inclusion goals. And so we were very excited to release a strategic plan that specifically outlined what the NMDC was going to do in terms of promoting diversity, equity, and inclusion. And so this is on our website. I would encourage you to take a look at what this strategic plan is, and we'll be looking at metrics associated with, are we doing a good job in terms of our mission towards these goals? And then lastly, I just wanted to highlight a couple new opportunities. So the first, we are launching as of today, this community conversations webinar series. We're planning for it to be over the course of fiscal year 22. Here is a snapshot of the next upcoming webinars that I think will be really exciting. We focused on not just what the NMDC is doing, but bringing in a broader range of experts who can speak towards the importance of ontologies or community engagement or bioinformatic workflows. And then I also wanted to mention that there is a new special collections through the journal Microbiome that is being launched, focused on integrative multi-omics approaches. And so I'm guest editor with Brian Headland, JP Dandore Aris, and Miriam Gazzardi, and we're actively recruiting manuscripts focused on the latest and greatest in multi-omics approaches, whether it's a lab work, experimental based, or bioinformatic based. So please keep an eye out if you're interested and have something ready that could be published in the journal Microbiome. And then I'll just leave here. We're a pilot project, but we're really excited in partnering with the research community to better enable fair microbiome data more broadly. And so I would encourage you, if this is of interest, please reach out, get involved, and I will end it there and happy to answer any questions.
Thank you, Emily. It's impressive to see the progress you've made over the couple of years. And it's exciting to learn more about this initiative. I encourage everyone to ask their questions in the chat window for Emily. And like you mentioned, probably like we are in the way of the lunch break or a coffee break for everybody. So I guess like we can take more questions on the Discord channel if there aren't any. And thank you, Emily, again for the informative talk. With that, I want to thank everyone who joined us today for another great round of talks. And I also want to thank all our speakers for wonderful talks this morning. If you have missed any of our sessions, the day one recordings are now available through the EMSL Learn website, and you can find the link to that in the chat window. And all the talks will be available through EMSL Learn website. I also encourage you to attend our virtual poster session that begins 12 noon Pacific time. The poster session will be in a separate Zoom meeting, and that link is also in the chat. During the post session, you will be able to join multiple breakout rooms and ask your questions from the poster presenters.